Wow, your food looks so delicious. Yeah, this is my breakfast today. Oh, I see. But do you know how our body digests this kind of food, which is the rice and the chicken as well that we consume?
Do you know what happened during normal condition? In a normal condition, when we start eating this kind of food, when our body is in a well-balanced state, the food that we eat, which consists of carbohydrate, lipid, and protein, will break down following their specific order. Carbohydrate metabolism. The complex carbohydrates are broken down into simple sugars like glucose, monomers such as sucrose and galactose. Glucose is central to cellular energy production, and when it is not used, it will store as glycogen in liver and muscles. Glucose will undergo glycolysis to form two molecules of pyruvate. Then, pyruvate will further degraded into acetyl-CoA, where it will continue degradation by the Krebs cycles and the following electron transport systems, which will form a bundle of ATPs, usually 36 in total. Lipid metabolism. How it is closely connected to carbohydrate metabolism? Fatty acid undergo beta oxidation and release one molecule of acetyl-CoA, which will further oxidize in Krebs cycle. Beta oxidation also produces NADH and FADH2 and directly enters the ETC, producing more ATP. Protein metabolism. Protein complex compound will be degraded into their simplest form, which is amino acid. Once it has been degraded into simplest form, the excess of amino acid will be used for energy production or will be converted into glucose or fatty acids. The metabolism of amino acid converge with glucose metabolism pathways.
Aisha, are you okay?
I'm having stomachache.
Why? You didn't take any breakfast this morning before coming to class?
No, I don't want to gain weight.
Aisha, that is wrong. Here, take this and eat this, okay?
Thank you.
You're welcome. Did you know that starve yourself is bad because?
So, what is starvation? Starvation is a severe deficiency in calorie energy intake where the level is below the needed for an organism's life for a prolonged period. It is based on the decrease in energy expenditure, saving of protein, carbohydrate, and utilization of lipids. During starvation, the metabolism rate slows, following the depletion source of energy and decrease of blood glucose level in our body. In order to be resistant to the changes, the body will develop an adaptation process which is known as negative feedback mechanism. During this period, it is called as homeostasis, in which our body will restore back to a normal state of glucose level and at the same time increase the production of energy. Thus, there are three ways that are involved during the negative feedback mechanism. The first one is carbohydrate metabolism, lipid metabolism, and protein metabolism. What happens to carbohydrate metabolisms during starvation? Glycogenolysis. In liver and muscle cells, glycogen will be converted to glucose 1-phosphate by glycogen phosphorylase with the help of glucagon and epinephrine. Glucose 1-phosphate then converted into glucose 6-phosphate. Glucose 6-phosphate can be converted directly to glucose by glucose 6-phosphatase or converted into fructose 6-phosphate first by phosphohexose isomerase, and the product will be glucose. Gluconeogenesis. In cytoplasm or mitochondria of the liver, pyruvate molecules must first be carboxylated by pyruvate carboxylase to produce oxaloacetate. NADH converts oxaloacetate to malate, allows it to leave the mitochondria. Malate is oxidized after exiting the mitochondria. The enzyme phosphoenolpyruvate carboxylase converts oxaloacetate to phosphoenolpyruvate. Phosphoenolpyruvate transformed into 2-phosphoglycerate by enolase. 2-phosphoenolpyruvate were catalyzed by phosphoglycerate mutase, where 1-phosphate group is transferred from 2-phosphoenolpyruvate to form 3-phosphoenolpyruvate.
3-phosphoenolpyruvate is catalyzed by phosphoglycerate kinase to form 1,2-bisphosphoglycerate, where one phosphate group is being transferred and an ADP is produced. Then 1,2-bisphosphoglycerate is transformed into glycerildehyde 3-phosphate by glycerildehyde phosphate dehydrogenase. Aldolase catalyzes the formation of fructose 1,6-bisphosphate from glyceride 3-phosphate. Fructose 1,6-bisphosphatase catalyzes the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. The enzyme phosphohexose isomerase converts fructose 6-phosphate to glucose 6-phosphate. The production of inorganic phosphate by glucose 6-phosphates results in the release of free glucose into the circulation. Glucose 6-phosphatase is the enzyme at work here.
But what about lipids? What happens to the lipid metabolism during starvation? In general, lipid will undergo gluconeogenesis, the breakdown of fats, triglycerides, where it consists of glycerol and free fatty acids, which will be delivered into the bloodstream to tissues. Glycerol will further undergo glycolysis, while fatty acid undergo beta oxidation. As I mentioned just now, glycerol will undergo glycolysis and produce acetyl-CoA. Fatty acid undergo beta oxidation. In each cycle of it will remove 2 carbon from fatty acid and release 1 molecule of acetyl-CoA. Beta oxidation also produce NADH and FADH2, and some fatty acids are also converted to ketone bodies. So, both glycerol and fatty acid will be converted to acetyl-CoA and insert the Krebs cycle. Did you guys know that ketone bodies is formed due to less sugar or less glucose in order to supply the body full need? Ketone bodies are basically nature's energy source during starvation. Well, what reaction occurs in beta oxidation? Beta oxidation occurs in 4 steps: oxidation, hydration, oxidation, and cleavage of acetyl-CoA. We start with acetyl-CoA molecules that present in mitochondrial matrix. Oxidation occurs which remove 2 hydrogen between carbons 2 and 3 and produce molecule called as trans-enoyl-CoA. Hydration then occur which add water across the double bond, and from this we will get beta-hydroxyacyl-CoA molecule. Oxidation then occur again, but the oxidizing agent at this time is NAD+ instead of FAD in the first oxidation. Beta-ketoacyl-CoA will then be cleaved down into 2 molecules, which is acyl-CoA and acetyl-CoA, where the new acyl-CoA is 2 carbons shorter than the previous one. Some extra information before we move on. Different length of fatty acid shows different energy yielding.
What happens to protein metabolism during starvation? Protein will be converted into amino acid, then it will convert to glucose to produce ATP. Amino acid that can be converted to glucose is called glucogenic amino acid that occurs through gluconeogenesis. Gluconeogenesis starts with deamination, which is the removal of an amino group from a molecule. Gluconeogenesis. Gluconeogenesis is a metabolic pathway that results in the generation of glucose from non-carbohydrate carbon substrates. Pyruvate converted into oxaloacetate with the help of pyruvate carboxylase and then being converted into phosphoenolpyruvate by PEP carboxynase. Then, it will continue with the reversible step of glycolysis. Glucose 6-phosphate is being converted into glucose by glucose 6-phosphatase.